Hello, everyone, and welcome to this panel discussion on adoption of new technologies in cell and gene therapy, development and manufacturing. We will discuss challenges and drivers and using AAV as example to just stay focused. I'm Natalia Markova. I will be the moderator of this panel. I am Business Development Director at Refeyn, which is an Oxford-based company developing mass photometry technology, which is increasingly finding applications in AAV workflows. Today's event is presented by LabRoots platform, and it is brought to you by Refeyn. I will, from the start, encourage you to post your questions to really not waste any time into the question section, which is Ask a Question box. Just type it there and click Submit. We will address your questions in the end of the panel. Also, if you have any issues with like sound or video, please type it into the Ask the Question box. So the topic of the panel is, of course, very broad and requires a broad experience. Therefore, I'm so excited to have this representative panel of great background experience. And I will start the introduction. With us, we have Brent Morsi, who is the principal consultant at Dark Horse Consulting. And Brent, you are supporting cell and gene therapy customers across all stages of the workflow, including CMC gap analysis, risk assessment, as well as interactions with regulatory. We also have Linda Randall from Pharmaron. She is Associate Vice President, Biologics. Really welcome to the panel. And Linda is leading development of innovative biologics spanning from proteins to viral vectors, and also very kind of broad stages of development from CMC to process validation and readiness for approval. Also welcome to Klaus Richter from Coriolis Pharma. Klaus is a group lead in AUC group at Coriolis, and you are developing analytical strategies and methods for characterization of drug substances and drug products. Very exciting. And Anthony Blashuk from United States Pharmacopeia, welcome. You lead initiatives to advance standards in cell and gene therapies, which is, of course, important to support high quality of the product as well as innovation for these products. And last but not the least, Sushmita Mimi Roy, who is the founder and principal at Ion Bio, and you are having projects in process analytical development of gene therapies and biologics from CMC to IND and approvals. So once again, welcome. And to start with in the discussion, Mimi, given your experience based on these projects on AAV development, for example, how close you think we are with a platform solution, analytics of AAVs for development and manufacturing?
That's a great question to start with. Thanks. I think we're coming to some conclusion on what CQAs are in AAV and what methods might be better to describe these and follow these. And it's wonderful to see platform analytical technologies with CDMOs, also with companies that have multiple gene therapy programs. However, I'm just coming off of the ASGCT Boston, which was a very exciting meeting to attend last week, and noticed that out of 2,000 or so presentations, 400 or more of them being talks, 900 presentations were on manufacturing and analytics. So there's obviously a lot of new results to present on how to process and make manufacture AAV better, and a whole plethora of new analytical technologies or new nuances to these technologies to measure these better, because the field suffers from the idea of the process is the product. And we're all scientists and we want to understand what are the important attributes of the product and how is the process going to be informed by analytics through the process and at the end in the DS and DP. So I do think there's a lot of creativity still in the field, and we are continuing to see new technologies. So it'll be important, it's wonderful to celebrate the progress, but I think it'll be important to continue to follow new highlights of analytical achievements.
Indeed. Thank you so much for this insight. Yeah, interesting. And I would say, indeed, the product and process understanding is still evolving. And in that sense, if we kind of look retrospectively at learnings and experiences with what it took to develop analytical platform for what is now called traditional biologics, like antibodies, for example. So how it worked and where we have to introduce advanced technologies in addition to what already is established. So what did we learn from established analytical platforms for antibodies, for example? And Brent, what could you say from your experiences looking back?
Yeah, I think the first thing to remember that, antibodies have been on a 35 or 40 year odyssey. So I think when we compare the state of cell and gene therapy to antibodies, I think we need to keep that in mind. Is they've been around a while. I mean, in the 80s, the early questions were, can we make these things safely? Can we have these drugs? Can we make them safely? Can we make them reproducibly? So the methods that were employed were fairly simple, simple ID methods, simple purity methods. In the 90s, we had a bit of an explosion in technical capability, and that allowed us to dig a little deeper in terms of purity, in terms of post-translational modification, and really be able to dig in. But then as we platformed these in the early 2000s, it led to a situation where we could identify risks, we could identify platform and product-specific risks. But the question was, what do we do with all this information, and what tests do we employ? And what tended to happen is that these became a lot of orthogonal methods, whether in characterization or release, and it became a very exhaustive type of situation where you were just throwing assays at this. As we moved into the biosimilar era, I think that's when we first started to use CQA-driven analytics. So the idea was we've made this rapid progress in analytics. We can measure differences, but what do they mean? And I think the biosimilar era really helped us, fine tune that edge of what analytical differences are clinically meaningful. And then in the 2020s, we matured from there, from orthogonal testing to more of a risk-based situation. And I think that's where we find ourselves at now in the antibody fields. But the question is, how do we port that onto the cell and gene therapy? And I think that's where the discussion will get interesting, is what solutions for antibodies are directly applicable and can be directly pulled into the cell and gene therapy space, what solutions don't really fit at all, and what solutions can be pulled over, but we have to modify either the way we approach it or with the way we think about it. So.
Yeah, indeed. Anyone else to bring thoughts to this discussion?
Yeah, indeed. I also feel like a lot of biophysical tools that we initially applied with the monoclonal antibodies and for biosimilar characterization had played their role there to mostly as a comparability measure, showing comparability of batches and these things. But then when we moved to the AAVs, it sorted out a little bit, and some of these methods indeed showed critical quality attributes, like, for example, the AUC, which had been around for 50, 80 years before, and now there was a specific question coming up in the AAV, like the empty field question, and all of a sudden that method, really old, had a specific place in this new field and was being very useful in the CGT field.
Yeah.
And I think that's also where a lot of creativity comes in, when all these methods now need to be compared, how good they perform in the specific AAV questions, and new methods come up, like the mass photometry, that needs to that show their place and show their value. And in the end, the biophysics still is very relevant, maybe with a little bit of a shift compared to the protein-based science before.
Yes. Yes, I see. And yeah, you mentioned that, yes, like this an effort bringing several methods together and trying to evaluate them. So I'm thinking of impact that United States Pharmacopeia is doing here also, looking at myself being at the stage when we were developing antibody methods and how much help we were getting. Anthony, can you share with us some overview from those days? What you probably also now taken over into cell and gene therapies.
Yeah, so I start out by saying one thing that's great about AAVs and cell and gene therapies, but also makes it more difficult to platform, is the uniqueness of all of these. So many of these are custom drugs for small patient sizes, and like every single genome is going to be different. So, on some aspects, it makes this capability to produce these amazing drugs, but platforming, especially like genome specific methods, is never going to be really feasible to some extent. You could do it for some common region, but you're always going to have to make that custom method for a very specific gene. That's never going to make it into a standard, just too many out there. So that's just one insight. But USPs, actually, we've been working on cell and gene therapy for over 20 years now. About close to 20 years ago, we came out with some chapters on ancillary material therapy, best practices for gene therapy, best practices for cell and tissue type therapies, but things have evolved since then. And about 5 years ago, we really saw what was coming up and decided to put a lot of focus on AAV. We saw that we saw all the trials going on for AAV, all the different companies working on AAV, and we saw a need for standards. And in the last 5 years, we've put a lot of focus in that. And it was really, in the last 5 years, been three key areas that we've really focused on. One of them is testing, and USP, NIST, and NIMBLE funded a large multi-lab study to look at a lot of different empty and full methods. I'm not going to get into that too much, but all the results from that were published. We compared the methods and really saw how they compared to each other. Another thing we started about 5 years ago, was develop an expert panel to draft a general chapter on AAV. That has already been published in PF, and that'll go official in the coming months. I wish I had the exact date on me, but I don't. But we gathered about a dozen experts from the field and drafted just a best practice anywhere, and that was from all the way from upstream all the way down to production, including stuff like how to choose the right formulation buffer, went over all the key CQAs and methods used for that. So it's designed for people new to the field, because there are so many small cell and gene therapy companies who need help getting started. But it's also intended for people who are familiar with the field, almost like a checklist, make sure you're doing everything right. And then lastly, in the last year plus, we've started really seeing a lot of physical standards, and these are going to be key to help companies do validation, method development, bridging, and we really try key CQAs from things that are upstream all the way downstream. And hopefully we have an opportunity to talk about these a little bit more later.
Yeah, thank you. And Linda, given that you are right in the projects where this is happening, basically, all the way to manufacturing, could you share experiences from Pharmaron, which methods you are using for AAVs, what works, and which methods maybe were borrowed, let's say, from antibody development as well, and what is missing? Oh, sorry, I think you are muted. Apologies. Okay, there are some. Okay, so while this is happening, as troubleshooting, Anthony, potentially, I don't think you mentioned that much on the reference standards that you are developing. So they are definitely helping in this type of technology comparisons and bridging. So what's new there, if you could update us?
Yeah, so as I said, in the last year, we've come out with a lot of different physical standards. We have about close to a dozen standards we've released. And what we've tried to do, and I want to take a step back too and mention that all our standards really are driven by stakeholder feedback. we go to conferences, we talk to people in industry, we talk to both CDMOs, CTOs, we talk to instrument manufacturers, and we really try to figure out what are the biggest hurdles and what physical standards can help support these. Mm-hmm. So based on all this feedback, we've decided to develop standards in 3 key areas, because essentially we found out that help was needed all the way across the whole manufacturing. So we developed a set of standards for upstream. Those are endonuclease, that's to help harmonize the activity testing for an endonuclease. Plasmids, set of plasmids with varying sizes to assess topology. We also have some impurity standards, because those are key quantitative methods that you need to look for at the end of manufacturing. Some of those include residual plasmid standards, as well as genomic DNA for both HEC293 and SF9. And then the last set is more like just final viral vector characterization standards, and those 3 are going to be the capsid titer standards, all different serotypes. That's one I'm really excited about. I actually had an opportunity on this where we're trying to harmonize capsid titer testing, because there's a lot of variability between different ELISA tests, and they do bluntly a pretty poor job of characterizing material that is used for a caliber, and just the nature of an ELISA is going to be dependent on that. And last, full standard. This is more our flagship one, where we felt there's a real need, going back to what we're talking about bridging and stuff, for a standard that's very well characterized by a lot of different methods. We've heard over the years from so many different people that they would purchase AAV from a commercial vendor, possibly even characterized as reference material, but it would have an empty full percent by one method. And then when they look at it in-house, they're getting a different value, and they don't know if the material's changing somehow. Is it the method that's different, or comparisons, or were they doing poor analytics? Was the vendor doing poor analytics? So what we did was we developed 2 AAV standards, one being an empty standard, one being a full, and we looked at it by a number of different methods. So on the COA alone, there's 6 different methods used to analyze it. Put in the spot: TEM, mass spectrometry, AUC, CDMS, UV, and SEC mols. I think I named all those. And for each one of those was done in a multi-lab study. So we have a collaborative study for all those, data from multiple labs. Certificate, we put the value for each of those different methods. And this is really helpful because it should play into everyone's hands. So if you're using one method, you have the value from that specific method. If you're looking to bridge from one method to the other, you have both of those methods. And having that standard in-house that will allow you to do that comparison, it can be really advantageous. In validation, that's another thing we see is difficult.
It is definitely a great help, and what a work with all these methods that you brought together to compare. So, and now Linda is back with the sound as well. And of course, you are in the environment where these type of samples, like references, are very important as well. Hopefully you're taking good use of it. So please share with us your experiences at Pharmaron. Thank you, Anthony.
Yeah, sure. Nothing like some technical issues to put the pressure on, is there? Or at least to prove it's live as well. So thank you for the patience. Yeah, so you were asking about the methods and platform methods and how we use them in Pharmaron. So we did set out to build up a platform of analytics. Started in 2016, building that with our platform for AAV. And it was important to us because we had a pipeline of products that we were developing at the time. But then when we moved over to Pharmaron in 5 years ago, serving as a CDMO, it's even more important to be able to access platform methods. Obviously, with the AAV, as Anthony and Brent have pointed out, it's difficult or challenging with any that are really product-specific assays. So it's really where we can use platform assays for the more generic, less product-specific assays, and establishing those, and then building up a platform validation that then we can leverage, so that for ourselves, it's all about trying to help develop the products quicker into the IND phase by accessing those platform methods with their platform validation, so that we can focus the analytical efforts more on developing the product-specific assays and those CQAs.
Yeah, thank you. And this is a nice leeway into product-specific CQAs, like empty full that was mentioned already, and focusing on this. So what are the evolving methods that are used to that end? And also, of course, we hear about partials and overfull, but let's start with empty full to start with.
Yeah,
would like
sure.
to, yeah.
Yeah, sure. So, yeah, the critical quality attributes, or CQAs, are quite distinct, aren't they, from the antibodies or other biologics? So the empty full and the aggregates, they're really the key CQAs, because they're directly impacting the potency, the safety, and the dose efficiency. So the full capsids are delivering the payload. Empty capsids are diluting that potency, really, aren't they, and can increase that immunogenic risk. So controlling the empty full is essential, both in clinical and commercial stages, and aggregates can impact the safety and the effective dose in exactly the same way on delivery. I suppose for me, over the last decade, really seen working on AAV products, that the analytical toolkit for empty full has really evolved quite significantly. But more importantly, I feel that it's the orthogonality that's really analysing those CQAs that's helped us, or starting to help us, unlock the product and process knowledge, so that we understand the products better, and then we can do a more comprehensive CMC analysis. So in terms of empty full testing, we usually use a combination of orthogonal methods. No single technique gives us the full picture, I don't think. AUC is the gold standard technique, because it gives us a much higher resolution of those separation of the empties, the fulls, but also the partial species, which are becoming increasingly more important to characterize. And it's also GMP compatible, so we've established the GMP capability and validated the assays so that we can use it in the QC release test panel. Obviously, that itself has a downside. It's quite labor intensive. It's technically a little demanding than perhaps some of the other techniques, and it requires a bigger sample volume. So I think some of the other techniques that have been mentioned, you know, the TEM or the cryo-EM, that gives you a good visual. Chromatography methods like anion exchange are becoming increasingly popular, and especially, I suppose, in the QC space, because they can be validated, they're more robust, so they give you a good pathway. And then we've also done a lot of work on the mass photometry, and that's increasingly being used for the process development and the QC. Ideally in the early phase, because it gives you really good information on your process and your product using small volumes. So, you know, we're using a fraction of the volume compared to the AUC, but we're getting good quality data and understanding of the product. But at the end of the day, I think for us, the AUC remains the gold standard.
Yeah, that's interesting, because we went through the same journey here. We also started out with comparing different methods that exist for doing the AAV. We started stability characterization with a nano DSF to limit sample amount, and then we moved into a published study on empty filled capsids, comparing a lot of different methods on which ones perform better, which ones give the same results, and which ones correspond to each other. So in the end, then after this journey in 2023, we decided to go for the validation qualification of the AUC, and for the qualification of course, the HPLC methods. so that we can offer them as QC release now. And indeed, it was quite a journey and quite a decision at that point to go for the qualification of the AUC, because it's quite an endeavor, lots of different things that one needs to qualify. The instrument itself, working with the vendor to make sure that supplier qualification is done well, looking at the software itself. And I think these steps were required quite some efforts to make in the end a good product of a qualified and validatable process. And this is, I guess, something that we also thought about for the mass photometry at that point. And we compared a lot mass photometry and AUC in different publications. And they work very well together. So we can bring them to the almost exactly same empty filled contents. The ion exchange chromatography the same. But for the qualification, we could not yet decide on going for a GMP of mass photometry. So the AUC is the one for us to address this critical quality attribute at the time.
Yeah, and you keep developing, because analytical strategies at Coriolis also.
Yes, we keep developing. We also like the platform approach, and I think AAV is perfect for the platform approach. We have different serotypes. But in the end, many attributes, they behave very similar. The methods need only a little bit of adaptation in some situations. So it's very good for a platform approach, and also to save sample amounts. I think it's important that we have a good starting method, because in many cases, especially early phases, the sample amount is limited. The sample is fairly expensive compared to the high-cons formulations that we get for antibodies. Sample amount is just lower, so we need to work with methods that consume only a small amount of samples. So we keep developing that portfolio, and look where need is there. Then I think there's a large panel of methods that can address questions, and exciting field at the moment.
That's a wonderful overview. Mimi, I'm looking at you, since you also have done quite a lot of work in these AAV focus projects. Please share what worked, what not, what you could see kind of going forward.
Sure. I was thinking about that a lot last week at the meeting, because many companies have moved away from ultracentrifugation to AEX chromatography as their polishing method. And it seems to me that just calling a peak empty or full can be a little misleading. And I think we need to think about orthogonal methods, but also what method is phase appropriate. So where I am at today is that I think NGS sequencing of the different fractions can be very, very helpful. I think ultracentrifugation really separates heavies and lights, but AEX doesn't separate heavies and lights. It separates capsids with different charges, and we can't assume that the 2 or 3 peaks we see, which don't use identification, for example, the mass spec method, CDMS, or mass photometry, or AUC, they don't tell us the identity. So I now think that using a lot of orthogonal methods, including identity, is very important early in the program. And once you've established a process, you don't expect these things to change much. It's really good to go towards a validatable method, a method that's robust, a method that's QC friendly. So you don't want to do CDMS in your QC lab, but you might be able to do AUC, which, not everyone has GMP level AUC, but people are developing it, as Linda shared. So I think it's more about product understanding in a very broad way early in the program, so that you understand that, for example, people are sharing now DNA leakage work, right? So DNA leaving the capsid using some fluorescence type methods and trying to understand, does the chromatography induce leakage of DNA? Are we moving from, more fulls to less fulls over the process? So understanding using a lot of orthogonal methods while developing the process, while developing the control strategy, while developing the final list of assays that are going to make the CQA list and make the release list is rather important.
Yeah, indeed. And there is this clinical relevance you talked about also, yes. So the methods report on parameters, on traits that are of significance to clinic. And with this, Anthony, thinking of what it takes to actually bring the methods to compendial level. So please tell us what you know about these efforts and challenges.
Yeah, it's something we're very actively working on because we think this would be a big benefit for everyone in the field. Just quick background for people who don't know, all our compendial less than 1,000 chapters in the USP are fully validated methods. We rely heavily on people to donate full validation packages to us. So if anyone has any validated methods they're willing to donate, here's a little offer to please reach out to me afterwards. But generally speaking, we receive these method donations of validated methods, and they ultimately go into the USP NF. And the benefit, as I mentioned, for these is once they are in the USP, they're recognized by most regulatory bodies. So companies don't have to go through the full validation. To do is a verification of that method for their specific product, which means they save all kinds of time, money, resources, and they can really speed up their production and save a lot of money. So we have a lot of chapter 129 is for MABs, a lot of compendial methods for that. We are very actively working on developing and acquiring compendial methods for AAV. There's some CQAs, such as potency, that just aren't going to ever make it there. They're going to be way too product specific, and having a platform method just isn't going to be a realistic option. Same with like genome titer. We talked about a compendial method for something like ITR, CMV, but ultimately the fact that everyone owns their GOI just not perfect that we.
Apologies.
Oh.
Okay, so some technical issue, but I'm sure Anthony will soon be back, and we'll
offer up that I think this is going to be a big advancement for the field, but also for the rare disease space. So one of the big challenges right now for rare diseases is they're pretty cost prohibitive in many situations, especially the ultra rares. And to the extent we can get USP or any pharmacopeia methods established, that could be the difference between having a product and not having a product. And we'd like to get to a situation where these N of ones become, it can become a little bit more reproducible, and it's not a heroic effort that needs to be put into these every single time, where it can, to the extent we can platform these and integrate these USP methods, I think it's going to benefit way beyond just the field of, the AAV field. It's going to benefit a lot of the rare disease space. So.
Mm-hmm. Yeah, absolutely. And I think also, as we discussed a bit earlier when we were setting the panel, this sort of initiatives of bringing flexibility to cell and gene therapy development, manufacturing, they are not on the expenses of quality. And the quality of the product is really coming with a lot of cost also for these rare diseases, indeed, for drugs direct into the rare diseases. So while we are looking for Anthony coming back, I was thinking that in the same direction to now look from the point of view of manufacturers and developers, and Linda, for example, like yourself. Oh, here okay, but anyway, sorry. Okay, apologies. Then we'll give some space for Anthony to finish on the compendial methods and the challenges and efforts. Please, Anthony.
Yeah, I'm sorry, company Wi-Fi strikes. But finishing my thought is that we are actually working on them. We are identifying ones that we think will be easiest to platform. Some ones that we're focusing on, like residual plasmid DNA, where most people are targeting antibiotic sites, or antibiotic resistant genes. So you can have same PCR targets for that. A lot of excipient concentrations and formulation buffers, such as like PS20. Even empty full, it's pretty much the same very similar method for a lot of these techniques, such as AUC and mass spectrometry. So we are actually working on those. We're prioritizing the ones that we think would be easiest to harmonize, and hopefully in the next year or two, we'll have at least a few and continue to build on that in our USP chapters.
Yeah, thank you. Yes, and then back to Linda. Yeah, so from the developers and manufacturers' point of view, so what requirements you see for the methods to be adopted, and that could kind of accelerate your development and manufacturing?
Yeah, I think one of them is what Mimi's picked up already. You know, in terms of in our labs, the empty full analysis gives the data on the proportion of the relative species. You know, so how many of empty, full, partial, but they don't really identify the genome. So as Mimi was talking about, I think we're focusing on developing the NGS capability to really interrogate those genomes and what's packaged within them. And we've found that the ability to use the long read NGS can determine the potential, the proportion of that full length DNA compared to partials. And so optimizing that is one area through which will really help us understand how to optimize the process. So taking it back into the process, understanding the product better, and the data we're getting will drive process improvements. And might even drive other things, you know, whether it's starting material, plasmid design, etc. I think the other area for us is we continue, and it's something that I think, Klaus, you picked up on earlier, is we're focusing very much on developing the small volume analytics, so that we're taking far less of the batch for not just the release testing, but the stability. So much of it is consumed for the analytical that your batch size for the clinical or ultimately the commercial becomes much smaller. So we're really looking at ways, and we've developed a number of methods that really focus down on the volume. So we're looking at and introduce the HIAC, for instance, subvisible particle testing, and that really gives us a reduction in the volume. So instead of 1.5 mil, we're much smaller than that. And applying UPLC where possible, again, to reduce the sample volume. So I think those are the sort of key areas where we're looking for new technologies to help drive how we work with AAVs and understand the data.
Yeah. Linda, that reminds me of another technology that I've been using a lot. We've talked a lot about DNA, you know, empties and fulls. The other CQAs of AAV are a lot around the protein chemistry, because the capsid is what the cells see first. The capsid is what gives an AAV its serotype and enables transduction in the first few steps as the capsid moves into the cells and into the nucleus. And again, as you mentioned, Linda, samples are so precious, and I found mass spectrometry to be incredibly useful, just as protein chemists use it for peptide mapping. I've been doing mass spectrometry of AAV capsids for over 10 or 12 years now, and following the PTMs quantitatively and understanding which parts of the capsid are critical to keep intact, to keep the potency at its maximum. And it's always interesting, especially being a consultant in CMC, to be hired by clients when they're at the PPQ stage and they might still be having serious manufacturing issues or issues with consistent product quality. And many times I find mass spectrometry to be very, very useful to understand what's going on in the bioreactor, or what's going on before and after chromatography step, using omics technologies, what's going on in the bioreactor using metabolomics, and also looking at the product and doing characterization of the peptides using peptide maps to see if they're acetylated enough, if they're not deamidated, or they're not getting phosphorylated or oxidized, or having some other inhibitory or negative PTM on them. So I really recommend folks to, again, do early analytics using these kind of technologies and have the process under control, so that you don't have to do mass spec, toward the phase 3 work, or trying to solve problems, or also doing any mass spec in the QC lab, which can be quite challenging. So using these more complex methods early on to get product and process understanding.
Yeah, thank you, Mimi. Anyone else to share their thoughts on this? So I think perhaps we can then, as we are coming to the final stage of this panel, could you share your outlooks for what you think deserves most attention in the future for AAV development and, of course, success in manufacturing stage? In terms of adoption of technologies, in terms of CQAs that are deserving more attention.
Yeah, I think I'm fully aligned there with Mimi, what she just said. We came quite a way now to get the structure and get the empty field situation also in a good way to analyze it, to describe it, and to understand the comparability of different batches there. But when it comes to the smaller changes that may influence the potency of the AAVs, like single amino acid modifications or so, then there could still be some surprises. And when Mimi describes now the mass spectrometry, how this could resolve it, would be an interesting addition to understand that. And I think many of the empty field things are well resolved now. The methods aligned, and we understand what to use for which purpose and under which conditions. And therefore, I think there's a big, maybe unknown yet to be found.
Yeah, thank you. Brent, perhaps your outlook.
So from my perspective, I think it's pushing forward with what I loosely call the genomic integrity aspects of this. So empty full is important, but we also need to know what's in there, if it's a partial genome, etc. And I think we're well on our way to implementing these. I think that's an area where we just need to push forward and keep going. Continuing on developing the reference material, I think will be huge, because of the variability and the problems transferring these types of assays. And finally, I would just plug in, we just talked about it briefly, but I think potency assays, and just in particular the choice of which readout that you want to employ for potency assays is going to take. I think that's something the field could spend a little more time on. I've had a lot of clients that get into late stage and really haven't thought about it enough, and that's in part because it's a combination of maybe not just taking something that works up front, but it's also there are some challenging situations that people find themselves in. They're targeting very unique cell type. They're targeting maybe a structural protein that doesn't really give a great readout. So I think all of that needs to be pushed forward to really help the field.
Yeah, thank you. Linda, anything you would like to add to the outlook?
Yeah, I think for me, we've got a lot of the techniques and the tools. Some of it's about how to apply it, like Mimi talked about in terms of the mass spec, how to apply it to the AAV. Now we have a better understanding from the other techniques. but it's also about, we can measure things, but I think it's about thinking about how does that data really drive us to improve our processes, introduce better control over our processes, more consistency, and ultimately, as Brent was saying, the link back to the clinic via the potency. So I think it's not just about what we test, but thinking how do we use that data to the maximum effect to drive forward the products and the processes.
Yeah. Thank you. Anthony, your thoughts on the outlooks?
Yeah, I think one thing that we're going to continue to evolve is the structure-function relationship. For AAV in general, we still know very little about that. Compared to something like MABs, where you know if there's a post-translational modification on one specific amino acid, how it's going to affect that drug product. We are far away from that. I think as our potency methods and mass spectrometry specifically continue to evolve, possibly even going into the MAM, multiple attribute monitoring for mass spectrometry, that could really be beneficial. Another thing I think that's going to really help analytics as well is the continued evolution of the AAV production. Unfortunately, so many analytical labs that here all the time just don't have enough material to essentially play around with. So as we continue to get higher yields and more labs can have more material just to play around with, take risk and develop new methods, that can help a lot as well. And in terms of the physical chemical technologies and the new instruments, I don't think that's going to slow down, especially so many of these are being able to be used for different cell and gene therapies such as LVV or LNPs. These instrument vendors want to continue to be the leader, so they're going to continue to push them and evolve their instrument capabilities.
Yeah, thank you. Well, I think we are coming to the end of the panel itself, but looking at the questions, what we have there. There was one I saw that when we were discussing CQAs and genome and significance. So there was a question on partials and overfull, how you see their significance in that sense, how much they can inform on product understanding.
That's an important topic. Okay, it's an important topic. I feel, especially when the inserts are shorter, we see below a certain threshold that multiple copies can jump into the capsid. Then we have a situation where there is like one or two of the short inserts in it. So I think that needs to be assessed. Also, the partials can be substantial for some, possibly depending on the packaging efficiency. And therefore, the methods, it is important that these methods can resolve also these other species. And it's not only an empty filled situation in many cases, but there is also a partial that should be at least understood how big it is. And possibly also this multiple copies of one genome inside is a topic that should be addressed and understood. AUC can do that, mass photometry can do it at some point. Also, for ion exchange, we had some problems resolving the intermediate species, the partials, but certainly methods are around that can address that. And it still will not show us what it is that is in the partials that needs the NGS, then I guess.
Yeah, that's a really interesting topic, Klaus. Can I say something about that? A little bit of a word of caution, I think, to users of these analytical methods. I see many times, particularly recently, the lumping of partials into fulls in the C of A. So many companies are just combining the results of those two and calling that their percent full. But just a reminder that anything with one ITR can get in and get packaged, but that doesn't mean it's going to be translated into functional protein in the patient. And we all know that full-length proteins survive, but translations of short-length or medium-length pieces of genes are subjected to proteolysis in vivo, and they may not be actually contributing to the clinical benefit. And so to really, as Klaus said, understand what is in the partials, how is it contributing to the potency, and is it really acting more as a full because it's going to give you some benefit clinically, or is it more acting as an empty? It's going to compete with the fulls to get into the cells and therefore actually decrease the effective concentration and effective potency of the product. So these are really, really important considerations, and they may not be the same for every program. They may be dependent on what the protein is doing in vivo. So, so rather than just kind of blindly putting these into convenient boxes so that we can feel good that we have 80% fulls or 90% fulls because that combined number looks better, I think we really need to be careful about how we understand these CQAs deeply and how we utilize it to bring solutions to patients that are safe and efficacious.
Yes, thank you. And there was also a question, Anthony, to you about new initiatives coming at the United States Pharmacopeia. So people are interested in how to apply, if there is anything of relevance to them. Maybe you can list something that is currently open for participants to come, I don't know, submitting their method or their samples, please, if you can comment on that.
Yeah, so there's tons of opportunities. We have constant expert panels. We just have a new expert committee those last 5 years, but we always have expert panels popping up. Keep an eye on our website. But we, even more direct, is we are constantly working with people asking for method donations, but we're also working on collaborative studies, whether that's testing one of our materials to provide value for a certificate, because as I mentioned earlier, our certificates all have multi-lab studies, and that's very reliant on collaborators. We also do individual testing or like R&D work with collaborators that can lead to publications or presentations. There's a lot of different ways. I would say, I think we have I don't have the email on top of me, biologics@USP. You can email that, or the easiest way is email me directly. I keep track of my email. First dot last name Anthony.Blazik. Email me directly, whatever you have in mind. I'd be happy to jump on a call and talk things over more and see how you can get involved and work together with USP.
Yeah, thank you. And yes, any more questions, please type into ask question and click submit. If you are watching this recorded, we can answer your questions afterwards. They will be still directed to us. I would very much thank everyone who were on the call, the audience, and of course the panel for this very nice discussion. And yeah, looking forward to progress in the field. Wishing you all the success with that. Thank you.